This week we're talking about consumer and forensic genomics, and in this video I'll be discussing SNPs. I've already mentioned this word a couple of times in class already. SNP, or SNP, we pronounce it SNP, stands for single nucleotide polymorphism. Single nucleotide here means that we're just talking about one base of DNA, one single position in the genome, a single A, a single T, a single C, or a single G that gets substituted to a different nucleotide in the DNA sequence. Polymorphism means that this SNP occurs in multiple forms in a population. That is, in some individuals at this one specific base of DNA, some individuals might have a T, whereas other individuals might have a C, for example. I do want to highlight that this is in a population. We really don't talk about SNPs in a single individual, although I can have two different DNA sequences at any position in my own genome, right? I can be heterozygous at any locus. However, that is not a SNP unless we're talking about those genetic differences in a population. Let's walk through an example. Say I have a population of C stars, and I'm interested in this one SNP that occurs at some particular position along the genome. So let's say, for example, that we have a DNA sequence on some chromosome that says A, T, G, C, T, A, G, and then there is a SNP in the population at this position that I've color-coded, where in some individuals, or on some genomes, I should say, that sequence is A, T, G, C, C, A, G. So some genomes have a T at this position and some have a C. This is what we'd call a SNP. It is a single site in the genome, and it is polymorphic. It comes in two forms. But again, importantly, it comes in two forms across a population, and it has to occur with some regularity in the population for us to designate it as a SNP. So instead of trying to write that entire DNA sequence in these different individuals as I create a population of C stars here, I'm going to color code them. I put two red dots in this C star to indicate that in this diploid organism's genome, it has the T version of this SNP, the T allele, at both positions across its homologous chromosomes. Because again, this organism is a diploid. It has two chromosomes that have the same sequence, and it happens to be homozygous for the T allele. Now, I'm going to start expanding this population of C stars. These C stars are making babies, this population is getting bigger, and at first, the genetic information at this one particular locus, at this T position, remains constant. Remember, mutations are random, they occur randomly, and that is how you would get a SNP in the first place, is through a random genetic mutation that changes that T to a C. And now I'm going to expand this population for a while, and I am not going to have a random mutation occur at this position. So right now we do not have a SNP. This entire population is homozygous for the T allele. Now as this population continues to get bigger, we see through a random mutation event the appearance of the C allele at that locus. So what happened is that one of the parents of this C star, either the parent who produced the small gametes or the parent who produced the large gametes, had a random mutation event that changed a T to a C at this one particular locus. There might be other mutations that have occurred in all of these individuals, but again, remember, we're only thinking about one particular locus in the C star's genome. Now this individual, the individual where the mutation occurred, is necessarily going to be heterozygous for that allele, right? Because it was only one parent that had the random mutation event, therefore it could only inherit that allele from one of its two parents. It's important for me to remind you here that this still is not yet a SNP. A SNP refers to a site that is polymorphic in a population, and right now we only see this other allele in a single individual. It is not occurring throughout the population. As generations and generations go on and new C stars are born and old C stars die, assuming that that variant did not cause significant harm to the organism, you may see it start to expand in the population. Although you should note that sometimes random mutations just die out in the next generation just by random chance. But in this case, let's say that generations later, our C star population now has 160 individuals in it, and you can pause the video here to check me, but I intended to make 32 dots here. So you can see that across our 160 individual C stars, which are diploid, we have that purple allele, which is a C position instead of a T for the red dot. It now exists in 32 positions across all of the chromosomes of the C stars. Again, I'm not saying that it exists in 32 C stars, but we see it 32 times if we're counting up all of the chromosomes. Now that we're seeing the C allele across the population, we can calculate the allele frequency of that allele. The allele frequency is the fraction of chromosomes that carry that allele, so the fraction of chromosomes in the population that have the C or the T at that position. And again, notice I'm not saying the fraction of individuals, because remember the individuals are diploid, they could have either one copy of the C or two copies of the C. So as I'm calculating the frequency of the C allele, I have to remember that I saw it 32 times. Some individuals were heterozygous, some were homozygous for the C allele, but overall, when I counted up all of the purple dots, I saw 32 of them, and that was in a population of 160 individuals. But those 160 individuals are diploid, so they're actually 320 or 160 times 2 chromosomes, if I'm thinking about the fraction of chromosomes that have the C allele. So in other words, I saw it 32 times out of 320 chromosomes, which is an allele frequency of 0.1 or 10% for the C allele. And now, for good measure, I can calculate the frequency of the T allele too. So you can go back to the previous slide that had all the C stars, or just trust me when I tell you that there were 288 red dots, or 288 times that I saw the T allele across those 160 diploid organisms for 288 times across 320 chromosomes, which is 0.9 or 90%. And here is a nice time for us to check our math and make sure this makes sense. Again, there are only two alleles in the population, T and C. So these frequencies of the C and the T allele should add up to 100%, because 100% of the individuals have either a T or a C. And what we see here is the frequency of C was 10% and the frequency of T was 90%. This frequency of 10% for the C allele is what makes us able to officially call this a SNP. SNPs have to be polymorphic in the population. They have to occur frequently enough for us to consider them as SNPs. So there are different thresholds for this, but you can think of about 1%, or sometimes people have a cutoff of 2% frequency in order to consider a variant a SNP. And that brings us to another important term that we use in genomics, which is single nucleotide variant. The single nucleotide bit still refers to the fact that this is one base of genomic information that's being changed, but the variant distinguishes it from a SNP, or single nucleotide polymorphism, because the variant just implies that it occurs in multiple forms and it doesn't make any assumptions about the frequency in the population. So when we first saw that C allele arise in the population, it was a single nucleotide variant, and it wasn't until it became frequent enough in the population that we considered it a SNP. A single nucleotide variant, an SNV, can occur even in your own body. You can have a mutation randomly occur in, say, a skin cell, and we would consider that a single nucleotide variant in you. Now, to connect this to our conversation about consumer genomics and forensic genomics, we have to compare some statistics about whole genome sequencing, which we were talking about last week, and what these consumer genomics services are providing. Now, today, in 2020, the relative cost of sequencing an entire human genome is about $1,000. But obviously, that's not what people are paying when they're doing 23andMe, right? These services cost about $99. So what is making up that difference in cost? Let's also remind ourselves about the size of the human genome. The human genome, the haploid human genome, which is what we'll consider here, is about 3 billion base pairs, or 3 gigabases of information, right? But what the consumer genomics services are offering is not that they're going to provide every base pair of sequence of your human genome, because they're not interested in that. They're only interested about differences in populations, differences in individuals, right? And not every site in those 3 billion base pairs of information is polymorphic. That is, we don't have a SNP at every population. So what they want to do is only look at SNPs. And in the human genome, we have about 3 million polymorphic sites. That is 3 million SNPs. But as far as I know, these services, they don't actually look at all 3 million of those even. They really just hone on the ones that show the most differences. So for 23andMe, I think they look at about 1 million of those SNPs. So that's why they are able to provide this information so cheaply, because they're not doing that whole genome sequencing, and they're not using the same technologies that we talked about last week either. The technology used by these companies isn't based on sequencing like we saw last week. They don't need to assemble a genome. They just need to see what variants are present or not present. And to do this, they use something that we call a SNP chip, or a DNA array. A SNP chip is essentially a microscope slide that has single-stranded pieces of DNA stuck to it. These single-stranded pieces of DNA are called probes. These probes are going to bind labeled target DNA. This labeled target DNA is what you are getting from your sample organism. So when you do 23andMe, for example, this is what they're getting out of your saliva. They're taking your DNA, chopping it up, and adding what I've drawn here as a star, a fluorescently labeled tag that is going to make it show up under the microscope. And now, this labeled target is going to bind the fixed probe DNA based on the patterns of complementary binding between bases to form double-stranded DNA. So once you allow those labeled targets to bind to the fixed probes, you would have a situation that I've sloppily drawn here, and you see that the sequence that was GTA binds this probe and fluoresces with its blue signal, and then the sequence that shows GCA binds this other probe and fluoresces with its purple signal. So overall, from this organism that I showed you, if I looked under the microscope, I would see the purple and the blue fluorescent dot, indicating that both alleles were present in my sample. So let's think about what would happen if we had a SNP chip for our C-star population that we worked on a little bit earlier. So I would have these fixed probes that have the DNA sequence matching the surrounding bit around the SNP, but also with an A to capture the T allele and a G to capture the C allele. And in an individual that is homozygous for the T allele, that is when I extract the DNA and prepare my fluorescently labeled DNA, it is only going to bind this one location on the SNP chip, right? Those sequences will only bind there. Nothing will bind the G position. So when I look under the microscope, all I see is a giant red dot indicating that that individual is homozygous for the T allele. Similarly, the same thing will happen with the individual that is homozygous for the C allele. I extract the DNA, I wash it over the SNP chip, and it binds to the probe that has the appropriate corresponding allele, and I look under the microscope and I see a giant purple dot. Now, for the individual that is heterozygous, on one chromosome it has the T allele of the SNP, and on the other chromosome it has the C allele of the SNP that has both DNA sequences present. So when I wash them over the probe, they will hybridize, and I will see not only the red dot, but also a purple dot too. So when I look under the microscope, I see both, and that lets me know that that individual is heterozygous at this SNP. Moving forward from here, we're going to see how we can use this information about SNPs to determine relatedness between individuals.